Welcome friends to a new session on genetics. In this session, we will discuss different types of traits related with sex, sex-linked inheritance, eye color in Drosophila, hemophilia in man, Hollandric inheritance, sex-limited and sex-influenced characters. Biological basis of heredity, sex-linked genes and their inheritance. Let us first see about types of sex-linked inheritance. There are 5 types of sex-linked inheritance. They are as follows: 1. X-linked inheritance, 2. Y-linked inheritance, 3. XY-linked inheritance, 4. Completely sex-linked inheritance, and 5. Incompletely sex-linked inheritance. We have already discussed X-linked and Y-linked inheritance in part 1. Other types of sex-linked inheritance are XY-linked inheritance, completely sex-linked inheritance and incompletely sex-linked inheritance. Now, we will see about XY-linked inheritance. Certain sex-linked genes are located on both X and Y chromosomes. They are called XY-linked genes and their inheritance is called XY-linked inheritance. XY-linked genes are partially linked because crossing over may occur in the homologous sections of X and Y chromosomes. Bobbed bristle is an excellent example for XY-linked inheritance in Drosophila. In humans, there are several defects of this type. Some of them are xeroderma pigmentosum, nephritis, retinitis pigmentosa, etc. are XY-linked. XY-linked inheritance in Drosophila is another aspect to be discussed. There is a gene pair in Drosophila which is controlled by genes that are located in the pairing region of X and Y chromosomes. This trait is known as bobbed, represented by small b, and shows shorter and slender bristles. When a wild type male is crossed with a bobbed female, all F1 progeny are wild type. However, if the cross is made between F1 bobbed female and wild male, all the F2 males are wild type and all the female progeny includes bob type and wild type in 1:1 ratio. Let us now see about xeroderma pigmentosum. It is a hereditary condition characterized by extreme sensitiveness to sun, especially ultraviolet radiation, leading to skin cancer. In some people with xeroderma pigmentosum, eye problems and neurological problems occurs, including learning disabilities, progressive hearing loss, loss of some reflexes, progressive neuromuscular degeneration and occasionally tumors in the central nervous system. The sensitivity to UV radiation results lighter skin pigmentation or very dry skin as seen in this figure, and a high risk of basal cell and squamous cell skin cancers and melanoma. Xeroderma pigmentosum is an autosomal recessive disorder in which mutation must occur in both copies of the gene, one inherited from father and one inherited from mother, in order to affect a person. This means that both parents must pass on a mutated gene to the child. A person carrying only one copy of the mutated gene is called a carrier. When both parents are carriers of a recessive mutation in the same gene, there is a 25% chance that a child will inherit 2 mutations and be affected, and this is clearly shown in this figure. The most common defect in xeroderma pigmentosum is the mutation of nucleotide excision repair enzymes, which leads to the reduction in or elimination of nucleotide excision repair. Unrepaired damage leads to mutation that alters the information of the DNA. This disorder may lead to cancer. Example, basal cell carcinoma, if mutations affect important genes such as proto-oncogenes or tumor suppressor genes. Now, let us see about nephritis. Nephritis is inflammation of the kidneys and may involve the glomeruli, tubules or interstitial tissue surrounding the glomeruli and tubules, and this is very clear in this figure. It can produce glomerular injury by disturbing the glomerular structure with inflammatory cell proliferations, which leads to reduced glomerular blood flow and urine output. Now, let us see about retinitis pigmentosa. It is an eye disorder that causes progressive vision loss. It affects the retina, the light sensitive tissue layer at the back of the eye, and vision loss occurs as the light sensing cells of the retina gradually deteriorate as seen in the figure. Researchers have identified several major types of retinitis pigmentosa based on their pattern of inheritance: autosomal dominant or autosomal recessive. Autosomal dominant retinitis pigmentosa is due to mutation in one gene which is on one pair of autosomes. In other words, autosomal dominant retinitis pigmentosa occurs when a fault in only one copy of a gene is sufficient to cause retinitis pigmentosa. A person with autosomal dominant retinitis pigmentosa has a 1 in 2 risk, that is 50% risk of passing on the condition to one of their children. This is because he or she will contribute one chromosome from each pair to a child. The chance of a child being affected is therefore 50-50 in each pregnancy, and this made clear in this figure. Depending on whether the chromosome carries the faulty gene or its normal counterpart, the child will be affected or not. Men and women are affected equally. Autosomal recessive retinitis pigmentosa, like autosomal dominant retinitis pigmentosa, affects men and women equally because the faulty genes are carried on the autosomes. To describe how this comes about, there are 4 possible outcomes if you toss 2 coins. Those are both coin heads, both coins tails, coin 1 heads and coin 2 tails, and coin 1 tails and coin 2 heads. Parents who are carriers have a 1 in 4 chance of having an affected child, that is 25%, and a 3 in 4 chance of having an unaffected child, that is 75%. There is a 50% chance of the child being a carrier, that is 2 in 4 chances. The cause of retinitis pigmentosa is the mutation of more than 60 genes. More than 20 of these genes are associated with the autosomal dominant retinitis pigmentosa. The most common cause of autosomal dominant retinitis pigmentosa is the mutations in the rhodopsin, that is capital RHO gene, accounting for 20 to 30% of all cases. At least 35 genes have been associated with the autosomal recessive retinitis pigmentosa. Mutations in USH2A, that is Usher syndrome 2A gene, are responsible for 10 to 15% of all cases of autosomal recessive retinitis pigmentosa. Females with an X-linked retinitis pigmentosa related allelic variant may be unaffected or may show clinical symptoms. Such affected females are usually or less severely affected than males of the same age. Now, let us see about completely sex linked inheritance. The genes located on non-homologous regions inherit together because crossing over does not occur in these regions. So, the genes located on non-homologous regions are called completely sex-linked genes, and the inheritance is called completely sex-linked inheritance. Example, hemophilia. The genes located on homologous regions of sex chromosomes do not inherit together because crossing over may occur in these regions. So, these genes are called incompletely sex-linked genes, and their mode of inheritance is called incompletely sex-linked inheritance. Example, retinitis pigmentosa. Let us examine about sex-limited characters. The idea of sex-limited genes was initially developed by Charles Darwin himself in 1871 in his book The Descent of Man and Selection in Relation to Sex. Sex-limited genes are genes that are present in both sexes of sexually reproducing species, but are expressed in only one sex and remain turned off in the other. Sex-limited genes are those which express characters in only one sex. The sex-limited genes may be located on any chromosomes. It is to be noted that sex-linked genes are located only on the sex chromosome. Their expression in the vertebrates is governed by the sex hormones. Sex-limited genes are responsible for the secondary sexual characteristics as well as primary sexual characteristics. Many studies have been published to explore the genetic basis of sex-limited genes. The sex-limited traits can arise in two ways. The alleles responsible for sexual dimorphism could begin by being expressed in male and female, then become modified, either promoted or repressed in one sex by regulatory elements or modifier genes, or can be limited to expression in only one sex when they appear first. Two interpretations are possible. One, the dimorphic alleles were initially expressed in male and female, then suppressed in females or became limited to males by regulatory regions that are completely dominant in hybrids. 2. The alleles were initially only expressed in males. The explanation for initial expression in both species is the involvement of cis dominance, where the factors that modify the gene are located next to the gene on the chromosome. This is in contrast to trans dominance, where mobile products that can affect distant genes are produced. These factors can be in the form of promoter regions, which can be either be activated or suppressed by hormones. These alleles come under regulatory control very quickly because none of the ornamentation seen in males is seen in the very next generation. These conclusions make it that at least some male specific genes due their expression by hormone levels, the absence of estrogen or the presence of testosterone. Examples: 1. In man, beard is produced by sex limited genes. A woman normally does not have a beard, yet she surely carries all the genes necessary to produce a beard, but the expression of that particular gene in ladies is prevented by the absence of a particular enzyme. But in rare cases, abnormalities in hormone secretion may occur in a woman which allow these genes to express themselves, and the result is a bearded lady. 2. Breast development is normally limited to women, but hormone unbalance may cause breast development in man. 3. The genes for deep masculine voice and masculine musculature in man will express themselves only when the male hormone is present. Genes for feminine musculature express themselves only in the absence of male hormone. They do not require the presence of the male hormone. A fourth case, in cattle, the milk production is controlled by sex limited genes. The bulls also carry the gene for milk production, but it is not expressed in the male sex. In cows, the genes for milk production makes its expression in the presence of female hormones. A fifth case, the brilliant plumage of peacock is also due to some sex limited genes. Sixth, an excellent example of sex limited inheritance is provided by the plumage pattern in birds. In domestic fowl of Leghorn, male have long curved fringed feathers on tail and neck, but feathers of females are shorter, straighter and without fringe. Thus the males are cock feathered and females are hen feathered. Results of various crosses show that hen feathering is a dominant character due to a dominant gene capital H, so that male as well as female with the genotypes HH or capital H small h will be hen feathered. However, cock feathering is due to its recessive allele small h, homozygous recessive condition small h is cock feathered in male but hen feathered in female, suggesting that in the presence of female hormone small h cannot express its cock feathering character. This was confirmed through experiments of gonadectomies, that is removal of gonad. When ovary of small h female was removed, it resulted into cock feathering. Similarly, in castrated males where testis was removed, expression of capital H is inhibited. It has been found that the expression of the genes capital H and small h depends upon the sex hormone. Any male that receives at least one dominant gene will be hen feathered, but those males which are homozygous for the recessive gene will show the cock feathered condition. Females are all hen feathered regardless of genotype, and this is very clearly shown in this figure. It shows that particular type of feathering depends upon specific type of genotype and sex hormones. The dominant gene H produces hen feathering depends upon specific sex hormone. The recessive gene produces cock feathering in the absence of female hormone and hen feathering in the presence of female hormone. Now let us examine about sex influenced characters. Some characters whose expression in a particular genotype depends on whether the individuals are male or female are known as sex influenced traits. The sex influenced genes are the genes in which the dominance is influenced by the sex of the bearer. They are located on the autosomes. The sex influenced genes express more frequently in one sex than in the other. The expression of such characters is not found on sex chromosomes, but they are under the influence of sex hormones and age. Not all phenotypic characters expressed in males or females show sex linked inheritance. For example, in humans, beard, mustache, masculinity are restricted to males. Similarly, masculinity, non-development of beard and mustache are restricted to females. These characters are developed at the adolescent stage and not at the childhood, and remain restricted to a particular sex. All such properties are called sex influenced secondary sexual characters. This can be explained with the help of example of horns in sheep. The pattern of horns is effectively controlled by a single gene, and other genes controlling this character are always homozygous. In Dorset sheep, both sexes are horned and homozygous dominant H+, H+. In Suffolk sheep, male and female are hornless and homozygous recessive, and this is clearly shown in this figure. When Dorset is crossed with Suffolk, the F1 are found to be hornless females and horned males. When F1 progenies wherein the genotype is H plus H, are intercrossed. In the resulting F2 progeny, males segregated as 3-horned and 1-hornless, while females segregated as 1-horned, 3-hornless. It is thus apparent that expression of only heterozygote is influenced by sex, H plus H being horned in male and hornless in female. In other words, while horned character is dominant in male, it is recessive in female. This influence is believed mainly due to female and male hormones. Certain characters in human beings which are not located on sex chromosomes are also believed to be sex influenced. For instance, white forelock, that is area of skin completely without pigment, absence of upper lateral incisor teeth, a type of enlargement of terminal joint fingers, hair lip, that is a fissure in upper lip, cleft palate, that is a fissure in roof of mouth, and stuttering, that is involuntary repeats of letters or words, are more common and more severely influenced in males. The baldness in man is another example for sex influenced character. This particular character is dominant in man and recessive in women. This is because the gene for baldness, capital B, the gene B in heterozygous condition expresses itself in male, but heterozygous females are normal even though they carry the gene for baldness, and you can clearly see this in this figure. It shows that only one gene is enough to produce a bald man, as a woman must require 2 such genes to be bald. In man, single gene for baldness can operate only in the presence of male hormone. There is another interesting sex influences gene in man which affects the length of the index finger. The short index finger is due to a gene which is dominant in the male and recessive in the female. Now let us summarize the foregoing discussions on sex limited and sex influenced traits. As sex chromosome determines the sex of an organism, the genes found on sex chromosome express their phenotype which is restricted to a particular sex of an organism. The XY linked inheritance, the X and Y genes are partially linked. It shows several defects in humans such as xeroderma pigmentosum, nephritis, retinitis pigmentosa, etc. The characters whose expression in a particular genotype depends on whether the individuals are male or female are known as sex influenced traits. The sex influenced genes express more frequently in one sex than in the other. Still, another class of traits called sex limited traits are distinguished by the fact that one form of this trait may be expressed in one sex only and not in the other. Sex limited genes are responsible for the secondary sexual characteristics. Now, I will give you certain assignments based on the discussions we had. 1. Describe XY linked inheritance. 2. What do you understand by completely sex linked inheritance? 3. Describe sex limited and sex influenced inheritance pattern with examples. 4. Write short notes on autosomal dominant or autosomal recessive retinitis pigmentosa. 5. Give brief account of plumage pattern in Leghorns. I will also give you certain references for your further reading. One is a book entitled Biology, 10th edition, published in 2015 and authored by Eldra, Charles, Diana and Linda. A second book is again entitled Biology, 8th edition. It is published in the year 2015 and authored by Eldra, Diana and Linda. The third reference is entitled Introduction to Genetic Analysis, 7th edition. The authors are Griffiths, Miller, Suzuki, Levantine and Gilbert, and it is published in the year 2000 and it is published from New York. A fourth reference is with the title Chromosomes and Heredity. It is published in the American Naturalist and the author is Morgan, published in the year 1910. The volume of this publication is 44, pages 449 to 496. A fifth reference is with the title Concepts of Genetics, 10th edition. It is authored by Klug and Cummings and published in the year 2011. A sixth reference is entitled Principles of Genetics. The authors are Gardner and Nusted and published in the year 1984. It is published by John Wiley, New York. I will also give you some websites for your further reference. One is www.genetics.org, www.theagricos.com, www.emedicine.medscape.com. We have come to the end of this session. We will meet in another session with a new topic. Until then, bye.